Hello, this is Soha Hassoun from Tufts University. I'm here to tell you about our work entitled Pathway Activity Likelihood Analysis and Metabolite Annotation for Untargeted Metabolomics using Probabilistic Modeling. This work is in collaboration with Professor Liping Liu and two of our graduate students, Ramtin Hosseini and Nida Hassanpour. A unique characteristic of metabolomics is its ability to reveal cellular functionality. We are already seeing some remarkable works of how metabolomics is allowing us to take a panel of biomarkers and making them clinically relevant or applicable for studying cellular processes. A recent example is a study published in Autism Research, where scientists at NeuroPoint DX, in collaboration with others, reported new findings from the Children's Autism Metabolome Project. The researchers identified unique metabolic signatures in over 50% of the children with autism in the study. This is an important step towards the goal of developing a panel of metabolomics tests that could form the basis for a biological screen to analyze the risk of autistic spectrum disorders. There are many other studies, including those focused on the microbiome and on exercise, that have shown metabolomics effective in identifying biomarkers and linking them to health and disease. Advancing metabolomics research to the point of broad use in healthcare and in other fields, such as biotechnology, will require significant improvements to the interpretation of metabolomics data. My presentation is about the interpretation of untargeted metabolomics data in the context of the biological sample. Given a set of metabolomics measurements and a network model corresponding to the biological sample, I'm interested in investigating two questions. Which metabolites are present in the sample, and which pathways are active? Here, we call a pathway active if the likelihood that the pathway generated the observed measurements is above a particular user-defined threshold. Our goal in this work is to answer these two questions using probabilistic modeling. Prior works on metabolite annotation mostly falls into one of two categories. Spectral database lookups allow comparing a measured spectra against those in a spectral database. However, spectral databases offer limited coverage. Metabolite annotation techniques such as CFMID, CSI finger ID, and METFRAG sift through a list of candidate molecules to identify the one whose spectral signature most likely matches to the measured spectral signature. Despite significant progress in this area, annotation rates remain low, where only a small fraction of measured data is annotated. There are several techniques that analyze the activities of pathways or modules within the network model. There is overrepresentation analysis. Here, we seek to know if a pathway is enriched in measured metabolites to a degree different than expected when compared to other cellular pathways or those in a reference sample. Enrichment analysis also encompasses techniques that compute the observed metabolite's centrality and connectivity. These metrics reflect the importance of a metabolite in a pathway or network. These earlier works focused on interpreting metabolomics in a differential fashion, for example, computing the enrichment ratio of a perturbed sample as compared to a reference sample. These techniques sometimes assume that the metabolite identities are known. One technique, called MummyChug, bypasses metabolite annotation and computes the functional enrichment of modules relevant to others in the sample. Unlike prior metabolomics analysis techniques that focus on differential analysis between a perturbed and a reference sample, in this work we focus on using all per-sample metabolomics data as well as the network model associated with the sample to interpret the metabolomics data. To better understand our data, we capitalize on our understanding of the uncertainty in mapping measurements to metabolites and metabolites to pathways. Here we have a snippet of a network that focuses on two pathways, Pathway 1 and Pathway 2. Each pathway is associated with several metabolites. Metabolites are represented as circles. While some metabolites, like J1, is associated with a single pathway, other metabolites, such as J2 and 3, are associated with more than one pathway. When performing metabolomics, we observed several masses. In the picture, masses are represented as squares. Some masses such as W1 can only be attributed to one metabolite, J1. Because J1 is only associated with the activity of pathway 2, the observed measurement W1 can be uniquely attributed to the activity of pathway 2. Other masses, such as the mass denoted by W2, can correspond to multiple metabolites, such as J2 and J3 here, which in turn can be associated with multiple pathways. We can all agree that we have more certainty in attributing W1 to the activity of pathway 2 than the certainty we have in attributing W2 to the activities of either pathway 1 or pathway 2 or both. The idea of this work is simple. We would like to use probability to model the uncertainties in mapping measurements to metabolites and metabolites to pathways. To solve this problem, we will use probabilistic modeling. Probabilistic modeling involves several steps. First, we define a model using random variables. Our model captures relationships between pathways, metabolites, and their masses. We then make assumptions about distributions on the random variables. Once we have a model and model parameters, we can then form hypotheses and calculate the probabilities of the hypothesis given the observed data. We could use Bayes' rule to compute such probabilities. To define our model, we introduce 4 types of random variables. The first set of variables are binary random variables that indicate if a pathway is active. A value of 1 indicates that the correspondent pathway is active, and a 0 indicates inactivity. There are as many random variables here as there are the number of pathways. The second random variable indicates if a metabolite was generated by a specific pathway. There is such a random variable for each metabolite within each pathway. The third set of random variables indicates if a metabolite is present in the sample. There are as many such random variables as there are metabolites in the network model. Finally, there is a set of random variables that indicates if a particular mass within a bin centered on each mass within the network is observed. There are as many such mass bins and corresponding random variables as there are masses in the model. Please note that this last random variable is the only observed random variable using the mass spectrum, while all other variables are latent or hidden. To define our probabilistic model, we assign distributions for the various random variables. We assume that pathway activities denoted by the letter A have a Bernoulli distribution with parameter lambda. That is, we assign each pathway a discrete probability distribution of a random variable which takes the value 1 with probability lambda and a value 0 with probability 1 minus lambda. Metabolites within each active pathway are equally likely to be generated by the pathway with probability mu. Z indicates this probability. Random variables associated with each metabolite in the sample, denoted by M, are derived based on the contribution of individual Z variables associated with each pathway metabolite pairing. Finally, random variables associated with each mass bin in the model is derived based on the observed accuracy gamma associated with the mass spectra, and if any metabolites associated with the corresponding mass bin was generated by an active pathway. So in this probabilistic modeling framework, our model parameters are lambda, mu, and gamma. We can assign the parameters some values. We have investigated the sensitivity of the model to these values. The model is not sensitive to these values as the structure of the model is the dominant factor that drives our inference results. The model we developed represents the joint probability distribution over all our random variables. Using this probabilistic model, we infer pathway activities and metabolite presence based on the observed mass measurements. Specifically, we calculate 2 probabilities. First, for each pathway, we calculate the posterior probability of the pathway being active. Next, for each metabolite in the model, we calculate the posterior probability of the metabolite being present in the sample. These metabolite probabilities can be used for annotation. If the calculated probabilities are above a user-defined threshold, then the probabilities are used to suggest and rank the best metabolite matches for each mass measurement. I'd like to emphasize that the way we used inference here is different from that used in prior works. For example, PROBMET assigns empirical formulas given potential formulas using inference. We carry out the inference using Gibbs sampling, which utilizes sampling to approximate our probability distributions. In this work, we use collapsed Gibbs sampling, as we marginalized out hidden variables Z and M. The sampler therefore only samples one random variable, variable A, associated with pathway activities. Significant speedups were possible when sampling fewer variables. We could run inference in 3 to 4 minutes when drawing 1,000 samples. Now that I have described our technique, I would like to share some of our results. We implemented PUMA using PYMC3, a Python package for Bayesian statistical modeling and probabilistic machine learning, focusing on Markov chain Monte Carlo and variational inference algorithms. We analyzed 3 datasets. The first dataset was a synthetic dataset that we generated with the goal of validating our probabilistic model. The second dataset was a metabolomics dataset that was collected by my collaborator, Kyung Lee from Tufts, for the Chinese hamster ovary cell. The CHO cell is a common model organism used in the pharma industry. We also analyzed a human urinary sample that was annotated by authors of that study. I will highlight some of the results. I'd like to first explain why we generated synthetic datasets. Despite the availability of datasets from several databases such as Metabolites or the Metabolomics Workbench, there are currently no untargeted metabolomics datasets with identified active pathways that could serve as the ground truth when evaluating our pathway activity prediction algorithms. The synthetic datasets were generated to mimic biological processes where genes within pathways work in concert and result in enzymatic activities that produce metabolites. As central metabolism and metabolic network topology is conserved across many organisms, we generated the synthetic datasets based on a representative organism. We selected the CHO cell as such a representative organism. Here's how the synthetic datasets were generated. A certain portion of pathways in the CHO cell were assumed active. We then assumed that a particular portion of metabolites within each active pathway are being produced by the pathway. We then assumed that we observe such metabolites using the mass spec and record their masses as our observations. For each portion of pathway and metabolite activity, we generate 100 datasets. We can then apply our inference algorithm to recover the process by which the observations were generated. We record the pathway activities and compare them against the activities that were used to generate the synthetic datasets. A pathway activity prediction algorithm will perform well if it accurately recovers the activities in the synthetic dataset. For the synthetic datasets, we compared the likelihood of pathway activities computed by PUMA against enrichment ratios. The enrichment ratio for a particular pathway is defined here as the ratio of measured masses that map to metabolites within the pathway to the number of metabolites associated with that pathway. We use the AUC, which is the area under the receiver operating characteristic curve, as our metric. A ROC curve plots the TPR, the true positive rate, versus FPR, the false positive rate, at different classification thresholds. The AUC effectively reports how well the method ranks an active pathway that is selected randomly more highly than an inactive pathway that is selected randomly. The datasets that are analyzed in this plot are generated assuming that 50% of pathways are active. We also show here different portions of metabolites within each pathways are activated. On the left side, we have the ROC curve for the PUMA. On the right side, we have the curve for the enrichment ratio. The AUCs are consistently higher for PUMA than for the enrichment ratios for different portions of active metabolites. For all our experiments, PUMA outperformed the enrichment ratio on average by 8%, with an average AUCs of 0.81 and 0.73 for PUMA and the enrichment ratios respectively. This tells us that PUMA is able to recover the pathway activity better than when using the enrichment ratio. I'd like to describe some metabolite annotation results that we obtained using PUMA. Our collaborators' lab had analyzed the CHO dataset thoroughly. After pre-processing the Metabolomics datasets, they provided Metlin and HMDB matches for each measurement when possible. They also used their tool Biocan, which aggregates measurements from spectral databases and different tools like MetFrag and from network connectivity. When using inference, PUMA assigns each network metabolite a likelihood of the metabolite being present in a sample. We considered assignments made by PUMA where the likelihood of the assignment was equal to or greater than 0.5. The top-ranked assignment was considered as the PUMA candidate set. Similarly, the top candidate set by Biocan was considered its candidate set. I'd like to share two important results when comparing Biocan and PUMA. The first result, which is summarized in the pie chart, shows how PUMA and Biocan predictions compare for 411 annotations made by PUMA. In general, there were lots of agreement or semi-agreement where there was some overlap between the Biocan candidate set and the PUMA candidate set. There were only very few cases of genuine disagreement where the two tools could not agree. There were 73 cases where PUMA suggested identities for measured masses and Biocan did not make any recommendations. Please note that PUMA utilizes only the mass of the parent ion while Biocan utilizes the spectral signature of the measurements. The second result is concerned with experimental validation. Biocan experimentally validated 50 of their predicted annotations against chemical standards. A subset of 37 out of 50 compounds were confirmed present based on the standards. Biocan's precision for this 50-compound dataset is 0.74. PUMA predicted 30 of the 50 metabolites as present in the sample. However, only 25 were true positives. PUMA's precision is therefore 0.833, thus achieving a significant additional 0.093 improvement in precision over Biocan. PUMA thus has high precision, returning substantially more relevant results than irrelevant ones. This work presents a probabilistic paradigm for metabolomics analysis. We can determine pathway activity and annotations from all the measurements and the network topology, all based on inference. We provide significant improvements over an aggregator tool such as Biocan. For more details, please read the detailed paper, which was published in Metabolites in 2020. The code is available on GitHub. Please give it a try. I'd like to thank the students in my lab and my collaborators, including Professor Lipping Liu, who was instrumental in developing this work. Thank you.